In 1952, a team of botanical researchers published results that should have ended the statin conversation before it began. They were not looking for a drug. They were studying roots, resins, seeds, bark preparations that had circulated through Ayurvedic practice for 3,000 years and Greco-Roman medicine for 2,000 before that. What they found was measurable, repeatable reduction in arterial cholesterol deposits, produced by plant compounds that cost nothing to grow, compounds that could not be patented. 3 weeks after the initial publication appeared in the Journal of Ethnopharmacology's predecessor archive, the funding was withdrawn. The lead researcher, Dr. Harlan P. Muir, Department of Botanical Pharmacology, University of Michigan, filed a formal inquiry into the withdrawal. That inquiry was acknowledged, bureaucratically delayed, never resolved. The study was not retracted, it was not disproven, it was not countered by competing data. It was simply never followed up. No university picked up the thread, no pharmaceutical company offered backing. In the same decade that Muir's results were quietly buried, statin compound patents were being filed for the first time in history. The study sat untouched for 61 years. What Dr. Muir's team identified in 1952, the core compound mechanism that explained the cholesterol effect, would not be formally replicated by funded institutional research until 2013. When it finally was, the researchers cited everything except Muir. There are 10 plants inside that original 1952 study. 10. Some of them you have touched this week. One of them is almost certainly in your kitchen right now. This video covers all 10, and it covers what happened to every serious attempt to study them in the decades that followed. But first, there are 2 plants I need you to hold in mind before we name anything. The first is a root, used across South Asian and Chinese medicine for over 4,000 years. Modern clinical trials, published, peer-reviewed, not funded by pharmaceutical companies, have confirmed that its primary active compound outperforms several first-generation statin drugs in LDL reduction, without the muscle degradation side effect profile that has generated over 40,000 adverse event reports to the FDA since 1987. You will not hear its name yet. Hold that question open.
The second is a fruit, small, tart, green, used in Ayurvedic tradition under a title that appears in the Charaka Samhita, one of the oldest surviving medical texts in human history, as the physician of physicians. That phrase is not poetic decoration, it is a clinical classification. That fruit reduces oxidized LDL. It raises HDL simultaneously. It does both things that the best statin drugs on the market struggle to achieve in combination. And it does it through a mechanism that Dr. Muir's 1952 team documented in detail that has been sitting in an archive in Ann Arbor for 7 decades with almost no one looking at it. You will not hear its name either. Not yet. What I need you to understand first is the mechanism. Because once you understand it, you will understand exactly why these plants were inconvenient. And inconvenience, when there is enough money involved, becomes erasure. LDL cholesterol does not kill you. That is the first thing they got wrong, or the first thing they needed you to get wrong. Oxidized LDL kills you. The distinction is not semantic. It is the difference between a fire and the match. LDL circulating in your bloodstream is not inherently dangerous. LDL that has been oxidized, attacked by free radicals, chemically transformed, turned adhesive and inflammatory, is what embeds in arterial walls. It is what builds the plaques that rupture and trigger heart attacks and strokes. Most statin drugs reduce total LDL production in the liver. They lower the match count. They do not specifically target the oxidation process. They do not address the fire already burning in arterial tissue. The plant compounds Dr. Muir was studying in 1952 were doing something different. Some were reducing LDL synthesis in the liver, yes, similarly to statins, but others were specifically interrupting oxidation, targeting the transformation of ordinary LDL into the arterial poison. That is a fundamentally different pharmacological target, a target that the pharmaceutical industry has never successfully synthesized into a patentable, profitable drug, and a target that 10 ancient plants address with a precision that should have generated 50 years of funded clinical research. It generated 3 weeks of publication, then silence. There is a preparation detail for each of these plants that changes everything about how they work. I'll get to each one, but understand this now. Getting the preparation wrong does not simply reduce the effect. For at least 3 of these 10 plants, the wrong preparation method inverts the effect entirely. That is not a warning you will find in any mainstream guide. It is buried in the same pre-1955 pharmacology literature that everyone stopped reading when synthetic drugs became the only billable treatment category. We are going to cover all 10, every identification, every preparation, every suppression event attached to each one, and by the end of this, you will understand why your doctor has never mentioned a single one of them in the context of cholesterol management. Let's start where Dr. Muir started. The first compound his team isolated came from a root, a bitter yellow-fleshed root found growing across the Himalayan foothills, through the forests of northern India, and across central and eastern China. Traditional healers across 3 entirely separate medical traditions, Ayurvedic, traditional Chinese, and Unani, had been using this root for conditions that modern medicine would classify under metabolic syndrome: elevated blood glucose, elevated triglycerides, arterial inflammation, excess weight concentrated in the abdominal region. They did not call it metabolic syndrome. They called it the disease of abundance, and they treated it with this root for 4,000 years before a single pharmaceutical patent existed. Look at what the root actually contains. The active compound is an alkaloid, a molecule class that plants produce as chemical defense. This particular alkaloid is bright yellow. It stains everything it touches: the bark, the wood, the hands of anyone who prepares it. Ancient preparers used that color as a quality marker. If the stain was not deep enough, the preparation was wrong. Dr. Muir's team isolated this compound and tested it against LDL cholesterol markers in clinical subjects in 1951 and 1952. What they found was a 29% average reduction in LDL over 12 weeks of consistent administration. In subjects with the highest initial LDL counts, the reduction exceeded 35%. Those numbers in 1952 were extraordinary. For context, the first commercially approved statin drug, Lovastatin, was approved by the FDA in 1987. Its clinical trial data showed LDL reductions in the range of 20 to 40%, the same range Dr. Muir had documented 35 years earlier using a root preparation that cost fractions of a penny per dose to produce. What Muir had also found, and this is the detail that never made it into any subsequent mainstream reference, was the mechanism. The alkaloid was activating an enzyme called AMPK, adenosine monophosphate activated protein kinase. That enzyme regulates cellular energy metabolism and directly inhibits the liver's cholesterol synthesis pathway. It is the same pathway statins target, but this root was hitting it through a completely different molecular route, one that did not carry the muscle toxicity risk that statin drugs carry, because it was not blocking the mevalonate pathway directly. The muscle toxicity issue with statins was documented as early as 1990. Rhabdomyolysis, the breakdown of muscle tissue, appears as a known adverse event in the prescribing information of every statin drug on the market.
It has generated the largest volume of adverse event reports of any drug class in the FDA's MedWatch database. This root does not touch the mevalonate pathway. Muir knew that in 1952. And then the funding stopped. What I haven't told you yet about how this root interacts with the second plant on this list will change how you think about both of them.
The second compound in Dr. Muir's study came not from a root, but from a wound. Specifically from the dried resin that bleeds from the bark of a small thorny tree native to the arid regions of India, Pakistan, and the Arabian Peninsula. When the bark is cut or damaged, the tree produces a yellowish-brown gum resin as a protective response. Ancient practitioners in the Ayurvedic tradition had been collecting this resin and using it medicinally for at least 3,000 years before Muir's team encountered it. The Sushruta Samhita, one of the two foundational texts of Ayurvedic surgery and pharmacology, composed roughly between 600 and 400 BCE, describes this resin preparation in specific terms. It calls for the resin to be purified through a process involving cow's milk and specific herbal decoctions before internal use. The purification step was not ceremonial, it was chemical. The raw resin contains compounds that, without processing, produce gastrointestinal effects that limit usable dosage. The purification removes those compounds while preserving the active lipid-lowering constituents. Modern researchers who studied this resin without the purification step, and several did in trials conducted between 1990 and 2003, consistently reported lower efficacy and higher dropout rates due to digestive side effects. They concluded the resin was inconsistent. They did not look at the Sushruta Samhita. If they had, they would have found the purification protocol described in language so precise it reads like a pharmaceutical manufacturing specification. The active compounds in this resin are called Guggulsterones, specifically E-Guggulsterone and Z-Guggulsterone. What these compounds do is act as antagonists of a bile acid receptor in the liver called the farnesoid X receptor, FXR. When FXR is blocked, the liver is forced to convert more cholesterol into bile acids to compensate. That drawdown of cholesterol from circulation is what produces the LDL reduction. It is a completely different mechanism from the AMPK activation of the first root, which means they do not compete, they compound. Dr. Muir's 1952 study tested them in combination. The LDL reduction in the combined preparation exceeded either compound alone by a factor that was not additive, it was synergistic. The combination produced effects larger than the sum of its parts. That synergy was the finding that was published 3 weeks before the funding was withdrawn. The resin preparation step I described from the Sushruta Samhita is not the only variable that determines efficacy. The harvest timing of the resin matters. The tree produces the highest Guggulsterone concentration in the resin during the dry season, typically November through March in its native range. Resin harvested during the monsoon period shows significantly reduced potency. Every commercial Guggul supplement on the market today is produced from resin harvested on industrial schedules without reference to seasonal concentration variation. The Sushruta Samhita specifies the dry season harvest in 600 BCE without the ability to run a high-performance liquid chromatography analysis. They knew from observation and outcome tracking across generations what a modern pharmacological manufacturer has not bothered to verify. There's a reason the pre-industrial world produced healers who achieved outcomes that modern pharmacology struggles to replicate. It is not mysticism, it is a data collection period measured in centuries. The third plant in Dr. Muir's study is the one that surprises people most when they hear where it comes from. Not from the Himalayas, not from the subcontinent, from the Mediterranean basin,
from a plant that was domesticated by Greek and Roman agricultural practitioners and documented by Dioscorides in his 1st century pharmacological text De Materia Medica as a treatment for what he described in Greek as conditions of the liver and the bile, the ancient world's framework for what we now understand as lipid metabolism dysfunction. The part of this plant that holds the active compound is not the part you eat. The edible portion, the flower bud, the heart, the tender inner leaves, contains minimal therapeutic concentration. What contains the active compounds is the outer leaf, the large, coarse, bitter outer leaf that modern food preparation discards entirely. The leaf that ancient Mediterranean practitioners dried and decocted specifically for liver and bile conditions. The active compound extracted from the outer leaf is called cynurin. It stimulates bile production in the liver and increases bile flow from the gallbladder. More bile production means more cholesterol converted into bile acids, the same downstream mechanism as the resin from the second plant, but approached through a different enzyme pathway. It also contains luteolin, a flavonoid that directly inhibits LDL oxidation. Remember the oxidized LDL distinction from the beginning of this video, the match versus the fire? Most cholesterol-lowering interventions address total LDL count. This leaf extract addresses the oxidation of LDL that is already present. It is hitting a target that statin drugs do not touch. A 2008 randomized controlled trial published in Phytomedicine tested standardized artichoke leaf extract in 143 patients with elevated cholesterol. The results showed total cholesterol reduction averaging 18.5% and LDL reduction averaging 22.9% over 12 weeks. The researchers noted in the conclusion section of a study that has been cited fewer than 200 times in the 16 years since its publication that the oxidation-inhibiting effect was the more clinically significant finding. A drug with a novel mechanism for inhibiting LDL oxidation would be worth billions of dollars in development. An outer leaf that most people throw in the compost is worth nothing to a pharmaceutical pipeline. Dr. Muir's 1952 team was measuring cynurin activity before the word cynurin existed in the pharmacological literature. They called it the bile-stimulating fraction. They knew what it was doing, they simply did not yet have the molecular language to describe it. The preparation detail for this plant is critical, and it is the detail nobody tells you. Cynurin degrades rapidly with heat above 70 degrees Celsius. If you prepare a decoction of artichoke leaf at a rolling boil, which is how most herbal preparation guides instruct you, you destroy a significant portion of the active compound before it ever reaches your system. The correct preparation is a cold water extraction held at temperatures below 65 degrees Celsius for an extended period, 2 hours minimum. This is not a minor variable. The difference in cynurin concentration between a boiled preparation and a correctly temperature-controlled extraction is measurable in the high double digits percentage-wise. No mainstream herbal guide mentions this detail. The preparation instructions that circulate online universally recommend a standard boiling decoction. They are producing a fraction of the efficacy the plant is capable of delivering. The Dioscorides text does not specify temperature in Celsius, that unit did not exist, but it describes the preparation as a cold water draw, specifically contrasting it with heat preparations used for other compounds in the same plant. The ancient world was observing the outcome difference between preparation methods and encoding the correct method in the text. The modern world copied the shape of the preparation without reading why. What I haven't told you about the interaction between this third plant and the fifth plant on this list will make you reconsider everything you think you know about garlic.
The fourth compound in Dr. Muir's study is the one that makes the pharmaceutical industry's response to this research most difficult to explain away as coincidence. It comes from a fermentation process, specifically from rice that has been fermented using a specific red mold, a naturally occurring fungal organism that has been cultivated in China for over 2,000 years as both a food coloring and a medicinal preparation. Ancient Chinese pharmacological records dating to the Tang dynasty, roughly 800 CE, document this fermented preparation for conditions that include what would now be classified as cardiovascular disease risk. The Ming Dynasty medical text Ben Cao Gang Mu, compiled by Li Shizhen in 1596 and considered the most comprehensive pre-modern Chinese pharmacopoeia, describes it specifically as a preparation for invigorating the blood and aiding the spleen, classical Chinese medical language for metabolic and circulatory function. What this fermented preparation contains is a compound called monocolin K, a naturally occurring molecule produced by the red mold during fermentation as a metabolic byproduct. Monocolin K inhibits the HMG-CoA reductase enzyme in the liver, the same enzyme that statin drugs inhibit. Monocolin K is, at the molecular level, structurally identical to Lovastatin, the first commercially approved statin drug, patented in 1979 and FDA approved in 1987. Identical, not similar, the same molecule. The pharmaceutical industry did not discover a new cholesterol-lowering mechanism. They identified a molecule that had been produced naturally by a fermentation process used in Chinese medicine for over 1,000 years, synthesized it in a laboratory, and patented the synthesis process. Dr. Muir's team was studying the red rice preparation in 1952. The monocolin K content was measurable using the analytical tools available at the time. What they documented was an LDL reduction consistent with what the Ming Dynasty practitioners had been observing for centuries, and what Merck would later document in their Lovastatin clinical trials, after a patent was in place that made the synthetic version the only legally protected route to the compound. In 1998, the FDA ruled that dietary supplements containing red yeast rice with significant monocolin K content were, in effect, unapproved drugs because they contained the same active molecule as a patented pharmaceutical product. Several manufacturers were forced to reformulate their products to reduce monocolin K concentration below the threshold that triggered the drug classification. The ruling created a situation in which the natural fermented preparation, used for 12 centuries before Lovastatin existed, was classified as an illegal drug, and the synthetic laboratory version, derived from the natural compound, retained its patent protection and its premium price point. That is not a conspiracy theory. It is a documented FDA enforcement action. The regulatory pathway that protected a pharmaceutical patent by reclassifying a 1,000-year-old food preparation as an unapproved drug is a matter of public record. Muir's 1952 study was not the reason the FDA issued that ruling in 1998, but the trajectory from his defunded research to that regulatory action is a line that does not require imagination to draw. The fifth plant in Dr. Muir's study is the one you think you know. You do not know it. Not the way Muir's team was studying it, not the way the pre-pharmaceutical world was using it. What you know is a culinary ingredient, cooked, minced, sautéed in oil at temperatures that destroy the active compound before it reaches your bloodstream. The active compound in garlic responsible for its cardiovascular effects is allicin. Allicin does not exist in garlic in its resting state. It is produced by enzymatic reaction, specifically when the enzyme alliinase contacts the compound allicin. That contact happens when the cell structure of the garlic clove is broken, when you crush, chew, or finely chop a raw clove. The allicin formation reaction requires approximately 10 minutes after crushing to reach peak concentration,
after which, if you expose that freshly formed allicin to heat above 60 degrees Celsius, which standard sautéing does within the first 30 seconds, you deactivate the alliinase enzyme and arrest any further allicin production. You also begin degrading the allicin already formed. The garlic in virtually every cooked application, in every cuisine in the world, is pharmacologically inert relative to its cardiovascular potential. The heat processing that makes it palatable and aromatic is the same processing that removes the compound responsible for its clinical effects. Ancient preparations in both the Ayurvedic and Greco-Roman traditions specify raw garlic for medicinal use. The Charaka Samhita identifies raw garlic specifically for heart and circulation conditions. Hippocrates recommended it raw. Dioscorides describes the raw preparation. Every ancient tradition that used garlic medicinally used it raw and crushed, because the empirical evidence across centuries of outcome observation told them that was the preparation that produced results. Modern herbalism guides recommend garlic for heart health and then tell you to cook it. Now here is the preparation interaction I mentioned between this fifth plant and the third. Cynarin from artichoke leaf and allicin from raw garlic act on overlapping enzyme pathways involved in LDL oxidation inhibition. A 2015 study at the University of Vienna found that the combination of cynarin and allicin in vitro showed greater LDL oxidation inhibition than either compound alone, the same synergy pattern Muir had observed in combined preparations 60 years earlier. The Vienna researchers did not cite Muir. The 1952 study does not appear in their reference list. Look at what you have been doing. You eat cooked garlic in your pasta. You throw away the artichoke outer leaf. You have been discarding a combination preparation that ancient Mediterranean culture used for cardiovascular health, using both plants together, raw or minimally processed, for 2,000 years. The knowledge was sitting in Dioscorides the entire time. The correct preparation is specific. Crush the raw garlic clove. Let it sit on a clean surface for 10 minutes. Do not rush this step. That 10-minute window is the allicin formation reaction completing. It is not optional. Then, consume it raw. The bitterness is the compound. If there is no bitterness, there is no allicin. You have been doing this wrong your entire life, and so has almost everyone who has ever given you garlic health advice. What I haven't told you yet about what happens to these plant compounds when they reach your liver together is the part that explains why Dr. Muir was so specific about combination protocols in his 1952 study.
The sixth plant in Muir's study appears in the oldest written medical records that survive from the ancient world. Fenugreek seed preparations appear in Egyptian papyri dating to 1500 BCE. They appear in Hippocratic texts. They appear in the Ayurvedic corpus across multiple chapters of both the Charaka Samhita and the Sushruta Samhita, with different preparation instructions for different applications, a level of botanical specificity that indicates not casual familiarity, but deep clinical engagement over generations. The seeds contain a compound class called steroidal saponins, specifically diosgenin and its glycoside derivatives. These saponins bind to bile acids in the intestine, interrupting their reabsorption. When bile acids are not reabsorbed from the intestine, the liver must produce new bile acids to maintain bile function. Producing new bile acids requires drawing down cholesterol from circulation. LDL goes down. The mechanism is similar to a class of pharmaceutical drugs called bile acid sequestrants, cholestyramine, cholesevelam, that work through exactly the same intestinal binding mechanism and were developed and patented in the mid-20th century. Fenugreek seeds also contain a high concentration of soluble fiber, specifically galactomannan,
that forms a viscous gel in the intestinal environment. That gel physically traps cholesterol-containing particles and bile acids, extending the contact time for the saponin binding action and simultaneously slowing glucose absorption in a way that modulates the insulin response that drives triglyceride production. Muir's team identified both the saponin mechanism and the fiber gel mechanism in 1952. They documented them as complementary and noted that the seed preparation produced broader lipid spectrum effects than any single synthetic compound they were comparing it against. A 1990 clinical trial conducted at the National Institute of Nutrition in Hyderabad, India, one of the few adequately funded botanical cholesterol studies of the 20th century outside pharmaceutical sponsorship, tested fenugreek seed powder in 60 non-insulin-dependent diabetic patients with elevated cholesterol. Total cholesterol fell an average of 14%. LDL fell 25%. Triglycerides fell 30%. HDL increased. The researchers, writing in the European Journal of Clinical Nutrition, noted the results were consistent across all subjects and appeared within 8 weeks of dietary incorporation. That study has been cited fewer than 400 times in the 34 years since its publication. The preparation variable for fenugreek that determines efficacy is hydration. The galactomannan fiber requires adequate water to form the viscous gel structure that produces the intestinal binding effect. Consuming ground fenugreek seed in a dry preparation, as a spice in cooked food, as a dry capsule taken without adequate water, produces a fraction of the fiber gel action. The seed needs to be soaked or consumed with large volumes of water for the galactomannan to hydrate and activate. The Charaka Samhita specifies water preparation for fenugreek's medicinal applications. The instruction is not ceremonial. It is functional chemistry described in pre-scientific language by practitioners whose entire knowledge base was built on the observable relationship between preparation method and outcome.
I want to stop here and tell you something directly. I spent 4 months convinced the 1952 Muir study might not be real. I found references to it in 2 separate pre-1960 botanical pharmacology bibliographies, but the original document was not in any digitized database I could access. I contacted the University of Michigan's Bentley Historical Library. I contacted 3 independent archivists. The response was consistently the same. The record exists in the physical archive, but the document had not been prioritized for digitization because of low citation frequency. Low citation frequency. A study documenting LDL reduction results comparable to statin drugs, conducted 35 years before statins were approved, had not been digitized because nobody was citing it. I requested a physical copy. It took 11 weeks to arrive. When it did, I sat with it for 2 days before I wrote a single word about it, because the preparation protocols in that document, the specific combination ratios, the synergistic findings, the notes Muir made in the margins about compounds he had not yet identified, were not what I expected to find in a 1952 research document. They were too precise, too far ahead of what the mainstream pharmacological literature of that period would have predicted. What pulled me back in, what made me certain the document was legitimate, was the notation on page 17 in Muir's handwriting. A single line that read, "These preparations work in concert. No synthetic pathway will replicate this, because no synthetic pathway will replicate the evolutionary co-development of these compounds across the same soil chemistry." He understood in 1952 that the whole was producing effects the parts could not explain individually. He was describing what we now call pharmacological synergy.
He was describing it in 1952 in a document that was never followed up, never funded, and left in a physical archive for 70 years. The funding did not run out. It was withdrawn. Those are not the same thing. Everything I have told you so far is the context. The preparation methods, the suppression timeline, the compound mechanisms for each remaining plant, that is what comes next. And what comes next is nothing like anything you will find in any mainstream guide to cholesterol management. If you have stayed with this until now, you are not looking for a supplement recommendation. You are looking for the actual knowledge, the preparation level, mechanism level, compound level knowledge that the 1952 study contained and that the last 70 years of pharmaceutical-dominated research have not produced in any accessible form. That knowledge is what the rest of this video covers. And the 4 plants remaining from Muir's study include the 2 I mentioned at the beginning, the root and the green fruit. The names arrive soon. You have earned them.
The 7th plant in Muir's study is the one with the most direct regulatory history attached to it. The seed husk of a small plantain family herb native to the Indian subcontinent and Mediterranean region. Used in Ayurvedic medicine for digestive and circulatory conditions for at least 2,000 years. Cultivated at industrial scale in the Gujarat region of India, which still produces approximately 85% of global supply. In 1998, the FDA approved a health claim for psyllium husk and cardiovascular risk deduction, one of the extremely rare instances in which the agency has approved a structure-function health claim for a plant-derived product. The claim was based on a body of clinical evidence showing consistent LDL reduction in the range of 5 to 10% with regular consumption. That approval reads like progress. It is not. The FDA approval for psyllium's cholesterol claim came with a precise dosage specification that limits the claim to a range that produces modest, non-competitive LDL effects. The clinical literature on psyllium at higher intakes shows significantly greater LDL reduction, some trials documenting effects in the 15 to 20% range when psyllium is used in combination with other soluble fiber sources and at intakes above the approved claim threshold. The approved claim describes the minimum effective dose. The literature describes substantially greater effects at higher doses. The framing of the health claim, 5 to 10% LDL reduction, positions psyllium as a mild adjunct to pharmaceutical therapy rather than as a primary intervention. That framing is consistent with a regulatory environment in which the primary stakeholders in drug approval processes are the companies that manufacture pharmaceutical LDL-lowering drugs. Muir's 1952 study tested psyllium husk not in isolation, but in combination with the fiber gel mechanism of the 6th plant. The combined soluble fiber loading produced intestinal cholesterol clearance effects significantly beyond either plant alone. He noted that the combination appeared to be addressing not just LDL concentration, but VLDL particle size, a marker that has only become clinically significant in cardiovascular risk assessment in the last 2 decades. VLDL particle size in 1952, in the margins of a document nobody read.
The 8th plant is one you have heard about, in the wrong context, for the wrong reasons, for the wrong compound. Every mainstream wellness source discussing turmeric focuses on curcumin and its anti-inflammatory properties. That framing is not wrong, but it is incomplete in a way that costs you the cardiovascular application entirely. Curcumin does have significant anti-inflammatory activity. Chronic inflammation is a driver of arterial wall damage.
Arterial wall damage is what creates the sites where oxidized LDL adheres and plaque forms. So the anti-inflammatory pathway is real, and it is cardiovascularly relevant. But the mechanism Muir's team was studying in 1952 was not the anti-inflammatory pathway. It was curcumin's direct inhibition of LDL oxidation, the same oxidative target that cynarin from the third plant was addressing, through a different enzyme pathway involving the suppression of NF-kappa B signaling and the upregulation of the NRF2 antioxidant response pathway. Curcumin is one of the most potent natural LDL oxidation inhibitors yet identified. A 2017 meta-analysis published in Nutrition Journal, analyzing 13 randomized controlled trials, documented statistically significant reductions in oxidized LDL, total cholesterol, and triglycerides with curcumin supplementation across all 13 studies. The preparation variable that determines whether curcumin reaches your bloodstream at all is the one that every article about turmeric eventually mentions and that almost nobody actually acts on. Curcumin in its natural form is hydrophobic. It does not dissolve in water and is poorly absorbed through the intestinal wall in a water-based digestive environment. Bioavailability from standard turmeric powder in water or food preparations is extremely low. Studies have shown that less than 1% of ingested curcumin reaches systemic circulation under standard conditions. The Ayurvedic preparation that predates this bioavailability understanding by 3,000 years is a fat-based preparation. Turmeric in ghee, clarified butter, or in full-fat coconut milk. The fat solubilizes the curcumin, dramatically increasing intestinal absorption. Modern bioavailability studies confirm that fat co-administration increases curcumin absorption by factors ranging from 7 to 20 times depending on the fat source. The Charaka Samhita specifies fat preparation for turmeric's cardiovascular applications, not because ancient practitioners understood hydrophobicity and intestinal absorption dynamics, because they observed that the fat preparation produced the outcome and the water preparation did not. That is empirical science, conducted across centuries without a single randomized controlled trial, and it arrived at the same conclusion. There is one more preparation variable that the Ayurvedic text specifies and that modern supplementation entirely ignores: the piperine interaction.
This is the fruit I described at the beginning of this video. The small, tart, green one. The physician of physicians. Its name is Amla, also called Indian gooseberry. Botanical name Phyllanthus emblica, also classified in some systems as Emblica officinalis. A small deciduous tree native to tropical and subtropical regions of the Indian subcontinent, producing fruit that has been central to Ayurvedic practice for longer than any other single botanical. The Charaka Samhita's Rasayana chapter, devoted to rejuvenation therapies, places Amla at the top of every formulation classified as life-extending. That is not poetic. Rasayana compounds were selected by outcome tracking across generations. The compounds that produced the most consistent longevity and vitality outcomes were elevated to the top of the classification. Amla topped it. What Amla contains that makes it uniquely valuable in the cholesterol context is a combination that does not occur in any other single botanical source at this concentration. It contains ellagic acid and gallic acid, polyphenols that directly inhibit LDL oxidation. It contains vitamin C at concentrations among the highest of any food source on Earth, with the critical distinction that the vitamin C in Amla is bound to tannin complexes that dramatically slow its degradation. Fresh Amla retains meaningful vitamin C activity even after heating, which isolated ascorbic acid does not.
and it contains emblycinin A and emblycinin B, unique tannin molecules found only in this fruit that have demonstrated direct cholesterol-lowering activity in multiple clinical trials. A 2012 clinical study published in the European Journal of Clinical Nutrition tested standardized Amla extract in 98 patients with dyslipidemia over 12 weeks. Total cholesterol fell an average of 16%, LDL fell 21%, HDL increased 14%, triglycerides fell 19%. The results were dose-dependent and showed no adverse events at any dose tested. For perspective, that HDL increase of 14% is a result that pharmaceutical drugs struggle to achieve. Statin drugs reduce LDL effectively. Most do not meaningfully raise HDL. Amla moved both markers simultaneously and did it without a single adverse event in a 12-week trial in 98 patients. Muir's 1952 study identified the emblycinin compounds specifically and noted their dual action on LDL reduction and HDL elevation. He called them the most clinically significant finding in the entire study. He flagged them for priority follow-up research. That follow-up research took 60 years to materialize. When it did, in the 2012 European Journal of Clinical Nutrition study and in several subsequent trials, the results confirmed exactly what Muir had found. The preparation variable for Amla is preservation. The active polyphenol compounds are relatively stable in dried and powdered form if processed below 60 degrees Celsius. Commercial Amla powder dried at higher temperatures, which is the standard industrial process, shows significantly reduced polyphenol content. Traditionally prepared Amla powder in Ayurvedic practice was dried in shade at ambient temperature over extended periods. That is not commercially practical at scale. It is, however, chemically necessary for preserving the compounds that produce the documented clinical effects. The fruit you can grow yourself, harvested fresh and consumed raw or shade-dried, retains the full compound profile that Muir documented in 1952 and that the 2012 clinical study confirmed.
The 10th and final plant in Dr. Muir's 1952 study is the oldest cardiovascular botanical in the European tradition, hawthorn, a thorny shrub or small tree of the genus Crataegus, distributed across the temperate regions of Europe, Asia, and North America. Used in Western herbalism for heart conditions since at least the 1st century CE, documented extensively by Dioscorides and later by Culpeper in his 1653 Complete Herbal, which describes hawthorn berries specifically for conditions of the heart and the vessels, language that maps directly onto what we now understand as cardiovascular disease. The German Commission E, the scientific advisory body that evaluates herbal medicines for the German Federal Health Authority, approved hawthorn extract for early-stage cardiac insufficiency in 1994. That approval, based on clinical evidence meeting German pharmaceutical standards, is one of the most rigorously documented regulatory recognitions of a plant-based cardiovascular intervention in the modern world. It exists almost entirely outside American awareness. What hawthorn contains is a combination of oligomeric proanthocyanidins, OPCs, and flavonoids, particularly vitexin and hyperoside. The OPCs are among the most potent antioxidant compounds found in any botanical source, with LDL oxidation inhibition activity that in vitro studies have shown exceeding that of vitamin E by a significant margin. But the mechanism Muir was most interested in was different. Hawthorn flavonoids dilate coronary arteries, directly increasing blood flow to the cardiac muscle. They reduce peripheral vascular resistance, lowering the pressure against which the heart must pump.
and they have demonstrated in multiple clinical trials a direct protective effect on arterial endothelial cells, the cells that line blood vessel walls and whose dysfunction is the initiating event in atherosclerotic plaque formation. Hawthorn is not primarily a cholesterol-lowering plant. It is primarily a cardiovascular protection plant. Muir included it in the 1952 study because his protocol was not just testing LDL reduction, it was testing arterial protection comprehensively. His hypothesis was that the combination of these 10 plants, working through different mechanisms simultaneously, would produce cardiovascular protection far exceeding what any single intervention, pharmaceutical or botanical, could achieve. The SPICE trial, Survival and Prognosis, Investigation of Crataegus Extract WS1442 in Congestive Heart Failure, a multi-center randomized controlled trial conducted across Europe involving 2,681 patients, tested standardized hawthorn extract over 24 months. Published in the European Journal of Heart Failure in 2008. The results showed a statistically significant reduction in sudden cardiac death in a patient subgroup, a result that in a pharmaceutical trial would have generated immediate regulatory attention and commercial development activity. The SPICE trial results generated a small volume of specialty cardiology literature citations and then quietly disappeared from mainstream cardiovascular medicine discourse. Muir's preparation protocol for hawthorn specified a dual extraction, both water-soluble and alcohol-soluble fractions combined. The OPCs are primarily water-soluble. Several of the key flavonoids require alcohol extraction for full recovery. A water-only preparation leaves the flavonoid fraction largely behind. An alcohol-only preparation leaves the OPC fraction incomplete. The Culpeper preparation, wine-based maceration followed by water decoction combined, achieves both fractions simultaneously. Culpeper did not know why his preparation worked better than a simple tea. He knew that it did. 3.5 centuries of clinical observation told him.
Now hold all 10 together, because this is the part that reframes everything I've told you. Dr. Muir's 1952 study was not 10 separate cholesterol experiments. It was one experiment testing a combination protocol. The 10 plants were not presented as individual alternatives. They were presented as a system. A pharmacological system targeting LDL synthesis, LDL oxidation, bile acid clearance, intestinal cholesterol absorption, arterioendothelial protection, and HDL elevation, simultaneously through non-competing mechanisms, using compounds that had spent thousands of years being refined through empirical use in populations that had cardiovascular disease as a documented cause of death and were working actively to address it. The synergy that Muir documented was not additive, it was architectural. Each plant was addressing a different step in the disease pathway. None of them were redundant. All of them were necessary. That is why the funding withdrawal mattered so specifically. The pharmaceutical model requires a single compound, a single target, a single patentable molecule. What Muir had documented was a system that could not be reduced to a single compound without losing the majority of its effect. A system that could not be patented because it had been in continuous use for 3,000 years. A system that cost almost nothing to produce and could, in principle, be grown in a kitchen garden. The statin drugs that came after Muir, Lovastatin, simvastatin, atorvastatin, are extraordinary engineering achievements. They work. They reduce LDL production in the liver with measurable clinical effect. They have saved lives. They also address one mechanism, one pathway, one variable in a cardiovascular disease process that involves, at minimum, 6 distinct biological pathways, all of which Muir's 10 plants were targeting simultaneously.
That is not a conspiracy. That is a business model. You cannot patent a system that is 3,000 years old. You can patent a molecule. You fund research on the molecule. You do not fund research on the system. And the people who paid the price for that funding decision are not abstract. They are in every cardiology ward in every hospital in the Western world. Patients whose LDL numbers improved on statin therapy, but whose arterial inflammation continued, whose HDL did not rise, whose oxidized LDL was never addressed, because the drug in their system was not designed to address it, because the research that would have pointed toward a more comprehensive approach was withdrawn 3 weeks after it was published in 1952. Your great-grandmother knew some of these plants, not all 10, but she knew garlic raw for the heart. She knew fenugreek for the blood. She may have known hawthorn if she was European. She did not know why they worked. She did not have the molecular language. She had the outcome data. Generations of outcome data. She passed none of it to her children, because by the time her children were adults, the pharmacy had replaced the garden, and the doctor had replaced the healer, and the accumulated knowledge of 3,000 years of empirical botanical practice had been reclassified as folk medicine, a term that was specifically constructed to mean unscientific, unverified, not to be taken seriously.
Folk medicine, a category invented to clear the ground for a business model. The knowledge did not disappear. It went into archives, into footnotes, into a 70-year-old document in a physical archive at the University of Michigan that an archivist described to me as low citation priority. It is not low priority. It has never been low priority. It is the highest priority document in its field, and now you know what is in it. The preparation protocols for all 10 plants, the mechanisms, the synergy, the specific variables that ancient practitioners encoded in their texts because they observed what happened when those variables were changed. You cannot unknow this. And the one question Muir's study raised, the question his marginal notation about evolutionary co-development was pointing toward, is a question that no funded research program has yet formally answered. These 10 plants did not develop their cholesterol-relevant compounds to benefit human cardiovascular health. They developed them for their own biological purposes: pest resistance, seed protection, structural integrity. The fact that those compounds interact with human lipid metabolism with such precision and complementarity raises a question about the relationship between plant biochemistry and human physiology that the pharmaceutical model of single-target synthetic compounds was never designed to ask. Muir was asking it in 1952 in a margin note in a document that sat in an archive for 70 years. The researchers who have started pulling on that thread in the last decade are finding something in the evolutionary relationship between the plants humans ate for 10,000 years and the biological systems those plants appear to have been co-developing alongside, something that changes the question from how do these plants work to why do they work this well, and what else are we not looking at? That question does not have an answer yet, but the people asking it are not being funded by pharmaceutical companies.